This video lesson is going to discuss protein synthesis with you. You will need to make sure you're writing these notes down in your journal. Write down everything that I write on the screen, as well as any illustrations that I draw. So follow along. Make sure you pause and rewatch any parts that you may find confusing. Before we talk about the specifics of how proteins are synthesized or made within the cell, let's first address the question, what is gene expression? Okay, so you've probably heard of this word gene before, right? Genes are sequences on our DNA strand that code for specific proteins. Okay, so when we are expressing a gene, we are, in a sense, we are making a protein that gene holds the instructions for. So this means we're going to go from our DNA sequence to RNA and ultimately to a protein that gets produced through this process. Okay, so protein synthesis then is the mechanism for gene expression. So protein synthesis is how we're going to go from our DNA sequences to producing an actual physical protein. Okay, the first step of protein synthesis is going to be transcription. Transcription is simply going to mean to make a copy of the DNA. So we copy the DNA. This is much like if you think about the word scribe, okay, where script comes from. The word scribe actually means to copy, okay? So back a long time ago, before we had copy machines and things like that, if you wanted something copied, you had to do it by hand. And those were the jobs of scribes. And so scribes literally would sit all day long and copy one book to another, okay, so that we could have multiple copies of the book. So transcription is our process of making a copy of the DNA. Okay, this copy of the DNA equals mRNA, okay, which we talked about in class together. mRNA is our messenger RNA, so it is our copy of our DNA sequence that we're going to be turning into, translating into a protein. Now, I want to point out the location of where this is happening. Okay, so this is occurring in the nucleus, okay, because this involves the DNA. So remember, DNA never leaves the nucleus, so transcription occurs in the nucleus. Okay, I also want you to notice transcription has a C after the S. Okay, this is going to be important. Remember that C is very close to the beginning of the alphabet. Okay, so when we talk about the second step of of protein synthesis, which is translation, okay, remember that they are in alphabetical order. So we do transcription first, and then we'll do translation second. All right, let's get into the details of how transcription occurs. Remember, this is occurring in the nucleus. All right, so the first step is going to be to unzip our DNA double helix to expose the bases, okay, so that we can add in the new nucleotides. So it starts out very similar to replication, but it's going to be different in the end. Okay, so remember in replication we had DNA polymerase. Well, in protein synthesis and transcription, we use an enzyme called RNA polymerase because instead of adding DNA nucleotides, we are adding or binding RNA nucleotides to the exposed bases. Now, this is going to follow a different base pairing rule. Okay, so remember, in DNA, adenine always paired with thymine. But in RNA, there is no thymine. Instead, thymine is replaced with uracil. So, when our RNA polymerase is doing its job and binding RNA nucleotides to the exposed bases, it's going to be matching adenine with a uracil, okay, and guanine will still get paired with cytosine. Okay, so before we go on, let's practice transcribing how RNA polymerase would transcribe a DNA sequence. So let's say I give you this sequence. This is our DNA template strand. Okay, A, T, G, C, C, A, G, T, A. Okay, there is our DNA sequence. Now, if we're going to figure out the mRNA sequence, or what RNA polymerase is going to add, how it's going to match to create the mRNA. So for mRNA, remember this A is now going to be paired with a U because there are no T's in RNA. The T will go with A, G with C, C with G, C with G, A with U, G with C, T with A, and A with U. Notice that in our completed mRNA strand, there are no T's. Okay, they're only U's for uracil. Okay, so this is how RNA polymerase is matching up these bases as it goes along and binds the new RNA nucleotide to the exposed DNA. All right, so our third step, after we have made our mRNA strand, our third step of transcription is we are going to edit the mRNA strand. Okay, because sometimes there are pieces in there that we don't actually need to send out of the nucleus to the ribosome. So when we edit the mRNA, we're going to have introns and exons. Okay, so an intron, this is a piece of the mRNA that are not important. Okay, so the introns are not important. This is like that extra bit of information that we do not need to send. Okay, it's not important part of the instructions. It's extra fluff. So I want you to think of this like introns, all right, stay in the nucleus. Okay, so these pieces are not going to leave the nucleus. Okay, we're going to remove these before we send out our final message. The other part are exons. So we have introns and exons. Exons, these are the important pieces. That get to exit the nucleus. So, okay, so think introns stay in, exons exit the nucleus. Right? Okay, so let's look back at this mRNA strand that we transcribed earlier. Okay, in this mRNA strand, let's say that we have an intron right here. This is an intron. Okay, so intron does not need to be leaving the nucleus. It's extra information. We're going to take it out. So in mRNA editing, we're going to take out the intron, and then we're going to piece together the rest of it. So UAC, CAU, all right? These would be our exons, okay, that are going to get to exit the nucleus. Now we have a special enzyme that actually removes this intron, okay, and puts together the exons. This enzyme is called spliceosome. Right? I'm not going to require that you know that, but I just want you to hear the name. It's called spliceosome. It's splicing together the pieces. Right? So the fourth and final step of transcription is that mRNA exits the nucleus and goes to the ribosome in the cytoplasm. So that is our fourth step. Okay, so let's draw this process of transcription. So here is our nucleus where this all is occurring. Okay, inside of our nucleus we're going to have our DNA molecule. So here is our DNA strand. Here, and we'll say this is A, T, G, C, C, G, T, A. Okay, so this is our DNA. In the first process of transcription, we're unzipping our DNA molecule. So it's getting unzipped, and so we have A, G, C, T, T, C, G, A. Okay, our enzyme that's going to be doing transcription is RNA polymerase. So this is RNA polymerase is going to move along our DNA code and add in the bases. Okay, so as it moves along this way, it's going to add in our bases, matching each one with its correct partner. So this T is going to be partnered with an A, this C with a G, this G with a C, and this T with a U. Okay, so this will be our mRNA strand that is being created. Okay. After this process, our mRNA is going to leave our nucleus and go over here to the ribosome, which is going to be our factory where we're going to actually produce our protein. And our DNA is then going to go back to the way it was before. Okay, so the DNA is left unchanged by this process. So that is an overview of what transcription looks like as it occurs inside the nucleus. Okay, so after transcription, we're going to be out in the cytoplasm, specifically on a ribosome. Our mRNA arrives at the ribosome and it's ready to be translated. Okay, so this is the second step of protein synthesis. Remember, I told you to remember transcription happens first because after the trans, there's a C. Well, in translation, there is an L, which comes after C in the alphabet. So you can remember translation happens second in protein synthesis. Some important things about translation include the fact that it occurs on the ribosome. Okay, now ribosomes can either be attached to our rough ER or free floating in the cytoplasm. And this is going to be, we translate mRNA into a protein. Okay, so this is like the factory reading the instructions, the blueprint, and building the product. Okay. All right, so the steps of translation are as follows. The first step is that our mRNA is going to bind to the ribosome. So it finds the ribosome and it attaches. Okay? The second step is that the mRNA is read 3 bases at a time. Okay, that equals 1 codon, right? While tRNA matches its anticodon to each codon and transfers the amino acid to the growing protein. I know that's quite a bit. So if you need to pause and write that down before you move on to the next slide, go ahead and do that now. Okay, so some terms that I used in that last slide about translation is that the word codon. Okay, so a codon is going to be 3 bases. So, for instance, A, U, G, that would be 1 codon. Okay, so 1 codon equals 3 bases. Okay, and 1 codon is going to code for only 1 amino acid. Right? And we have a couple of very, very special codons that I want you to have written down and have memorized. Okay, so we have our start codon. This tells our ribosome where to start translating. Our start codon happens to be AUG. Okay, and this codes for the amino acid methionine, or MET. Okay, that's going to be start. Right? We have some stop codons because our ribosome needs to know where to stop translating, right, when it's done. These are going to be UAA, UAG, and UGA. When the ribosome comes across any of these 3 codons, it's going to know that it's time to stop translating and that the protein molecule is done and it's finished. Okay, so those are 4 important codons I want you to know. All right, before we practice some translation, let's draw an overview of what this looks like as it's occurring inside of our cell. So here's our ribosome. Our ribosome has a large subunit and a small subunit. Okay, it's kind of like a hamburger bun almost. Okay, and what's going to happen is our mRNA is going to fit right in the space between where our 2 buns, where like the meat would go in between the 2 buns. Okay, so let's say our mRNA sequence is AUG and CAU. Right, there's our mRNA sequence. All right, what's going to have to happen is, remember, this is reading it 3 bases at a time. So we're going to read these 3 together and these 3 together. Now, we have these tRNAs that are going to be carrying the amino acids. So our tRNAs are going to be coming in. It looks like an upside down T, right? And it's going to be it has an anticodon, which is the complement of our codon. So these are our codons. Remember, codon here, anticodon. You should be able to tell me that the anticodon is found on the tRNA, and the codon is found on the mRNA. So again, this is our tRNA molecule. Okay, so our codon is complementary to our anticodon. So U goes with A goes with U, C goes with G. All right, up here at the top, this is where it's carrying its little amino acid, right? Which in this case happens to be methionine because it's our start codon, right? So this tRNA comes in, matches its codon, and then it heads on out, but it leaves its amino acid here. Okay, as we come in, as our next tRNA comes in, it's going to be carrying a different amino acid. So this one is going to come in. This one's going to have the complement to the AU, which would be GUA. So this one's going to come in and match up over here, and it's going to be carrying a different amino acid. So once we get these 2, once these match, these are going to be connected. Okay, they're actually going to be a lot closer because this is actually a moving situation. It's not stationary. And as we go along, this mRNA is fed through here. These tRNAs come through the top. As it matches, the amino acids are added, and we get eventually a growing protein chain that's going to have a very special purpose inside the cell. Okay, so we are not ribosomes, but we are able to translate what an mRNA sequence is into the amino acid sequence, which would then tell us which protein it is. Okay, and so to do that, we have to use what I have here on the screen, which is called a codon chart. Okay, you're going to be getting one of these tomorrow to put in your journal, and you're going to be very familiar with using this by the end of the week. So if I give you this sequence, this mRNA sequence equals AUG, GCC, UAG. Okay, we have to figure out, okay, what is this going to be translated into? What amino acid sequence? And so to do that, remember our ribosome reads in 3 bases at a time. Okay, that's one codon is 3 bases. So the first step I want you to do is I want you to actually go through and just draw a line every 3 bases so that you can easily see what you're looking for on the chart. Okay, once we've done that, now we're going to start looking over here on our chart. You can notice right here on the left side is the first letter. Okay, so down along this left side is where you're looking for the first letter of the codon. Okay, across the top, these are going to be our second letters, and along the right side is our third letter. So for instance, if we wanted to look up AUG, we're going to come down here and we see that A is in our third row. So there's where A is. So we know we're going to be somewhere in this row. Somewhere in this row is where we are going to be looking to find our answer. Okay, now we know that our second letter is U. So for our second letter, we look across the top and we say, oh, here's our U. It's in our first column, so that means now we can narrow it down even further. We know we're going to be somewhere in this first box. Okay? Now for the last letter, we can look over to the far right side and we say our third letter. So looking here, we have UCAG. Our last letter is G, so that means our answer is, it's our start codon, or methionine would be our first one. Okay, so I want you to take a second and see if you can look up GCC and UAG. Alright, you should have gotten methionine as your first, is what we did together. Then for GCC, you should have gotten alanine. And for the last one, you should have gotten a stop codon. Right? We're going to be doing lots of practice on going from DNA to mRNA to translating. So let's do one last example in our notes, and then you will be done with your video. So I'm going to give you a DNA sequence. Here's our DNA sequence. Our DNA sequence is going to be T, A, C, G, A, T, T, A, T, C, A, G, right? Let's figure out our mRNA sequence. Our mRNA sequence then would be, remember T is going to pair with A pairs with U, C pairs with G, G pairs with C, A pairs with U, T pairs with A pairs with U, T pairs with A, C pairs with G, A pairs with U, and G pairs with C. So there's our mRNA. Okay, now I'm going to ask you to figure out the tRNA anticodons. Now we don't actually use our anticodons to look up on our codon chart, but I want you to get in the habit of figuring these out. Okay, so A is going to pair with T, T with A, C with, oh wait, I made a mistake. Are there any T's in RNA? No. So A is going to pair with U, U with A, C with G with C, C with G, A, U, U, A, U, C, A, G. Okay. Now our last step is going to be to figure out our amino acid sequence. Okay, now to do this, you are looking only at the mRNA sequence, not the tRNA, so don't get confused. So we're going to draw our lines where our codons are, and we're going to look these up. Now you should already have memorized that AUG stands for start, okay, which is methionine, right? CUA, so let's look back over here, CUA, so we have C, okay, so let's look back over here. We have C, U, A. So C and U intersect in this box, and A is going to be here. So our next one is going to be leucine. Then we have A, U, A, U, A, which would be isoleucine, which is abbreviated ILE. And then we have GUC. So we have G, U in this column, and C, which would be valine, as our amino acid sequence. Okay, I want you to keep this fresh in your brain so that you can do this on your warm-up tomorrow. Make sure you have everything written down. Go back and rewatch any parts that you are confused on, and that is all.